Justin Turner, and today we're going to be discussing the ethical debate that surrounds the CRISPR cas9 topic. Alright, so let's begin. So just to start off, this is a brief overview. So based on the consequentialism theory of ethics, it analyzes the outcomes of a situation and determines if the good outcomes ultimately outweigh the bad. And we're going to be looking at both, and ultimately it determines that the CRISPR genetic editing is actually ethical in most regards. So during the presentation, we're going to examine the history of gene editing, the 4S framework, then the possible applications of different treatments, how far to extend it, and when to start treatments, when it is ethical to do so. So just to get some background information, the CRISPR cas9 cluster regulator. So cas9 stands for clustered regulatory interspace short join repeats. Essentially, this is DNA that are found in different genes, and there's repeated sections throughout. That's what gives different traits. And the cas9 is actually an enzyme that puts the DNA at specific locations. And then over on the right is a picture of how this actually happens. So there's a targeted sequence of DNA, and then essentially that DNA is cut. The cas9 then allows, which is the enzyme, surrounds it, and then other DNA can be put in its place. And this is kind of how we can modify genes all of a sudden very quickly. So there is precedence for this. The CRISPR cas9 is really beneficial for humans. We have been able to do this with plants in the past, and this is really just a sped up way of the traditional method, which is natural selection. Farmers always have done this throughout history of selecting the best plants and animals to breed with in order to produce the strongest results. However, today with our growing population, it is essential for this process to actually be sped up and not just be a random mutation that is then carried through generations. It's important to be able to select that first try and to get it right. This will increase productivity in crops. A big success is the golden rice, which has allowed for more vitamins to be put into the rice in order to be more beneficial. So the CRISPR actually allows for the stability in the inheritance process and not just have it be kind of a random effect that happens. We really only understand it though through one generation. So now we go into the 4S framework, which kind of evaluates the ethics behind this, and it's 4 different steps. So it involves the safety of this process, the significant harm being reduced in the that's actually helpful, and then the succeeding generations and how they receive these benefits, and then also the social consequences of editing genes. Alright, so we're going to start with safety. Basically, it involves clinical studies in humans. There isn't really a precedence for long-standing genetic editing. However, the table on the right has the 3 different ways that we are able to genetically engineer our DNA. So the first 2 are the ones that are currently, so the ZNF and the CAL, both other methods. The one on the far right is CRISPR, and as we can see, it is the least complex in design, and it is the easiest to manipulate and kind of choose which sections of DNA are going to be split. So the safety is actually very, we know what's going to happen at the end. While there is off-target effects, they are less frequent than the other 2, but they are more varied, which is where that safety concern comes in, especially in human testing. So the significance of harm is really the benefits that come from benefits of can result from the CRISPR genetic editing. Really, the point of this is to be a new therapy for diseases. These diseases are often ones that don't have any cures now, such as cancer, such as HIV, there's plenty of blood disorders and other genetic disorders such as fibrosis, muscular dystrophy, and even Huntington's can be spotted and stopped with the CRISPR method. Another thing that needs to be looked at is the benefits for our climate that's changing. Humans now need a way to adapt more quickly. As temperatures rise and the heat-related mortality, it's important that we're able to counteract this in some way, and this would be one of the methods we would be able to do that. So really, the position of where the negatives of genetic editing come from is in the succeeding generation. When do we stop genetically editing ourselves and our future generation? It's very different from plants to humans. For plants, we're looking to increase their productivity and we can always change things. They're not what we consider living beings that have feelings like that. As humans, we're physically changing a person and we don't know the full effects of it. So that's where kind of that ethical debate comes from. So really it's the designer baby debate of whether or not we can go into embryos and alter things from the beginning. Can we change eye color? Can we ultimately do things like that?
So moving on from the designer babies, there's also other social consequences that would be involved in genetically editing ourselves. A big thing would be in minorities where there is a difference in genes. Really, we're only studying it in one type of person, and that's more of the white male genes, and we're not sure. There hasn't been research into how it would affect other groups of people. Also, there's a geographic limitation. Where is this going to be accessible to? And how would it reach everybody that would be affected by the different diseases that it could help cure? And how would we get to those people? So this could ultimately widen the gap in social classes of who it's available to, who can afford it, how things like that would come about. And ultimately, this would lead into more discrimination based on genetics. And that's something we really don't want. So big thing is the transparency needs to be a necessity. The National Human Genome Research Center is working very hard to make sure that everyone would have access. Research is becoming more apparent to and for everyone, and it's not just going to be for that one specific group of person. Right? So really the opposition side is that it is unprecedented in humans, especially with editing, just that first generation. Their argument is more of it goes against nature, even though we are able to select the person that, people that do have kids. Clinical studies can be dangerous, especially since we're not sure how the difference between going from plants to animals to humans really works and the differences between them. Also, when it's consensual, it's one thing for it to be used as a therapy for a disease that's incurable, and it's another thing for it to be embryos that haven't even been born yet. And then also the accessibility is also a big thing with this social factor involved. However, the biggest argument behind all of this is that humanity is more defined by our culture than our genes, and a lot of these can be argued against that when people band together and making it more accessible, making it, setting guidelines of when it's available and when it's not, ensuring that it's safe through clinical studies, and all of that, and that really as long as discrimination is going to happen and as long as we can stop that, that's really where that's going to come from and really be the strongest argument against any of the opposing sides. So with consequentialism, there are pros and cons to genetic editing. A lot of it revolves around that it's just not at that point yet. to be full encompass what it could do. But the parts that involve helping with diseases that currently don't have cures far outweigh any of the risks that would be involved in possibly mutating other aspects of DNA that wouldn't be there anyways. Right? So here are the references if you're looking to learn more about the CRISPR method or anything involved. Thank you. Have a good night.